So welcome to Chapter 3. In this chapter, we're going to discuss genes, DNA, the chromosomes, and cellular division. Expression of genes. How actually are the genes expressed within the cells? What is a dominant gene? This is in what state will it be expressed in the phenotype with a dominant gene? What is a recessive gene? And in what state will it be expressed in the phenotype? What is a codominant gene? What is an incomplete dominance? What is an X-linked or sex-linked trait? What is an allele? And what is the locus? So these are questions that you should be able to answer at the end of this chapter. Also, you should be able to tell me what the genotype and what the phenotype are. So, how many X chromosomes are necessary in the sex chromosomes of humans, or actually any animal? Well, Mary Frances Lyon actually came up with a theory that stated that only one X is actually necessary to carry all of the genes of the sex chromosomes within an organism. And so what she theorized was the formation of something known as the Barr body or sex chromosome. And oftentimes you can actually see this in a neutrophil, and it's this little splotch right here off of the nucleus. This is actually an inactivated X of the double X in a female. And so since we only need one X, and that's a good thing since males only carry one X, then the extra X is simply inactivated in the cells of the female. Chromosome analysis, and it certainly is not this. Okay? How do we actually look at chromosomes? Well, division of cells is actually stopped in metaphase of mitosis, and the cells are treated so that they will swell. Each human cell has 46 chromosomes, each consisting of two sister chromatids joined at a centromere. Then this extracted chromosomes will actually be stained in order to be able to see them. And oftentimes the chromosomes then are arranged in a standard pattern known as a karyotype. So this actually is just simply the chromosomes from a single cell arrested in mitosis. And the chromosomes were extracted and stained so that you would be able to see them. And you can see that they actually are kind of this X shape, and this is because two sister chromatids are actually held together here at the centromere. And you'll notice that some of them are very short while some of them are very long. When we do a karyotype, we arrange the chromosomes from the longest to the shortest of the autosomes, and then the last pair is going to be the sex chromosomes. So this is the result of a human chromosome painting in which each pair of the chromosome is stained a different color by hybridizing with specific fluorescent probes. So each pair can be identified by both structure and color. So in other words, this one and this one are a pair, and it looks like probably this one and this one are a pair, or maybe this one and this one, but it's kind of hard to just see in this whole mess of things. So by doing the karyotype, now we can actually see them aligned up with each other and in the correct place. So from the longest to the shortest of the autosomes, and then the sex chromosomes will be the last one on the karyotype. Now this is a human karyotype because there are 23 pair or 46 chromosomes. And what is the sex, the genetic sex of this individual? So here's just another normal karyotype, and we actually will look at each pair to see if there's any abnormalities, and so far nothing, nothing, nothing. And again, then we look at the sex chromosomes to find out the genetic gender of this individual. So what is the genetic gender of this individual? What is the genetic gender of this individual? Now on this one, there's an abnormality. Can anyone see it? Well, as we look at the autosomes, everybody looks normal as we go down through all of the pairs. But when we get to the sex chromosome, oops, we have a problem. Instead of having a having a pair, we have a triplet, and we have an X, and a Y. So first of all, can you tell me the genetic gender of this individual? But is this a chromosomal abnormality? And it actually is. And we would call this actually XXY 47, or the term or the name of this disorder is actually Klinefelter syndrome. And we'll talk about that later on. So what about anything here? Well, the X and Y look just fine, and all of the autosomes look fine, except for, oops, there's chromosome 21, instead of being a doublet, is a triplet, and this is actually Down syndrome. Now, are all genomes actually the same? And they're not. They vary in number of chromosomes and number of base pairs and number of genes. So just as an example, the Drosophila, the cute little fruit fly that most of us absolutely hate, unless we're a geneticist, they have 8 chromosomes, is the diploid number or 2 in number. So in other words, they have 8 or 4 pair. Corn has 20 chromosomes. Giraffes only have 30 chromosomes as they're 2 in number. The common earthworm has 36 chromosomes. Cats have 38 chromosomes, but dogs actually have 78 chromosomes. So this is what I say, dogs rule, cats drool, right? Okay. Corn has 20 chromosomes, soybeans actually have 40 chromosomes. Man has 46 chromosomes, and the great apes actually have 48. Elephants have 46 chromosomes, while cattle have 60. Potatoes have 48 chromosomes, so are they related to the great apes? Well, no, because they have different number of genes. Donkeys and horses are slightly different. Donkeys have 62 chromosomes, horses have 64. But in horses and donkeys, we can actually create a hybrid between the two. And that hybrid is actually mule. The mule actually has 63 chromosomes. Because you can't divide 63 evenly, the mule is actually a hybrid that is sterile. And then this pretty little butterfly has 380 chromosomes. And then this plant actually has 1,260 chromosomes. So the number of chromosomes vary among the different species, as well as the number of genes and the number of base pairs. So chromosomes are where we keep the genes, and this picture is just simply showing the banding of the genes. So each one of the dark bands is actually a gene on this chromosome. The genes are the segments of the DNA that determine cell property. They are the code for a particular cellular product, a protein, whose will account for the structure and the functions within the cell. The genes are the basic unit of heredity. They exist in pairs normally in eukaryotic cells or alleles. Each one of the alleles came from one of the parents. So within each allele, one in each chromosome, and each allele will occupy or have specific sites known as the locus where identical genes will be located. So if I have the other one of this pair, the gene located right here on this one and on the other one will code for the same trait. Now, the alleles may not code for the same type of trait. In other words, you might have one allele for blue eyes and one for brown eyes, but both alleles code for eye color. And they're paired in the same way as chromosomes, except in the sex cell of the sperm and ovum. If both alleles are the same or code for the exact same characteristic, then we call it call it homozygous. If the alleles are different, we call it heterozygous. Double coils of DNA are combined with histone proteins in eukaryotic cells. This is not true in the prokaryotic cells. The chromosomes are present in the nucleus and control cell activities. Now we do have some extranuclear chromosomes, and that's in the mitochondria and the chloroplasts. But these chromosomes are actually not inheritable in the way that the nuclear chromosomes are. Simply the mitochondrial and chloroplastic DNA will be carried in the mitochondria and chloroplast during cytokinesis. The nuclear DNA is what is copied during reproduction. So in eukaryotic cells, they exist in pairs, or what's known as the 2N or diploid number. Each one of the alleles, one is derived from the male parent and the other one from the female parent. In humans, we have 22 pairs of autosomes, which are similar in size, shape, and appearance. The chromosome pair number 1 is the longest, chromosome pair number 22 is the shortest, and these actually will code for all of the characteristics except for gender. The sex chromosomes, the pair 23 in humans, determine the genetic gender of the individual. The X chromosome does carry around 1,000 genes that code for other things, but the XX and XY is what will determine genetic gender. So the DNA is a double helix, and then the helix itself is wrapped around the series of histone proteins, of which there are actually 8 major types. Of the 8 major types, they form then these cube-shaped structures in which we wrap the double helix around. And when the chromosome is in its relaxed state of chromatin, we actually will be able to see these kind of ball-like structures called nucleosomes, which are these single cubes of histones wrapped with the double helix. But when we get ready for reproduction, these coils will actually coil up and coil up, forming supercoils, and this actually then packages the DNA very tightly in the chromosome form. Chromosomes are only seen when the cell is undergoing cellular division. Otherwise, it's in the more relaxed state of chromatin so that we can access the genes within the DNA. DNA is made up of a polynucleotide chain. Each nucleotide is made up of a pentose sugar, which is deoxyribose, a phosphate molecule, and one of 4 nitrogenous bases. And hopefully this is mostly review. The pyrimidine, cytosine, and thymine, and the purines, adenine, and guanine. Who pairs with whom across the double helix? Well, we'll find that the nucleotide of the sugar, phosphate, and nitrogenous base, the nitrogenous base sequence will determine the amino acid sequence within proteins during protein synthesis. This double helix model was first proposed by these two gentlemen, Drs. Watson and Crick, back in 1953. In the year 2000, we mapped the human genome, and we'll talk a little bit about that later on. It was interesting to find out later that actually Watson and Crick had developed their theory of the structure of DNA based upon another researcher in another group named Rosalind Franklin. She actually came up with this double helix model in 1951, and Watson and Crick finally gave Rosalind Franklin credit in the 70s. Now, during replication of DNA for cellular reproduction, first of all, we have to untwist and unzip the double DNA strand. Each strand then will act as a template to create an exact copy. Complementary base pairing will be accomplished by an enzyme called DNA polymerase. And there are actually several of these, and we're not going to go into specifics on which type, but just know DNA polymerases make DNA. Adenine pairs with thymine, cytosine pairs with guanine across the double helix. The reason for this is adenine and thymine form 2 hydrogen bonds, whereas cytosine and guanine will form 3 hydrogen bonds. A number of enzymes are necessary for DNA replication, and copying the DNA for cellular reproduction will occur prior to mitosis and meiosis. Copying the DNA to messenger RNA will also be accomplished where we only copy one strand of the DNA to messenger RNA, and this is for protein synthesis. So, some important enzymes in DNA synthesis and transcription of DNA to messenger RNA. First of all, DNA gyrase. DNA gyrase, also known as a topoisomerase, relaxes the supercoiling ahead of the split known as the replication fork. Basically, what DNA gyrase does is it untwists the double helix, laying down the parallel strands of DNA kind of like a railroad track. Helicase is what actually unzips the double-stranded DNA. So while this says it unwinds the double-stranded DNA, helicase literally separates the 2 strands at the replication fork. DNA ligase will make covalent bonds to join the sugar phosphate backbone in DNA strands. And this is in any DNA strand. It's primarily, though, used to join Okazaki fragments and new segments in excision repair. DNA polymerases synthesize DNA, but they also have a function to proofread and repair the DNA as they are matching those complementary base pairs. Endonucleases and exonucleases will cut the DNA backbone and for different repair and insertions, and we won't really spend a lot of time with these two. RNA primase will make an RNA primer off of the DNA template. The reason that we need a short segment of RNA to start DNA synthesis is because DNA polymerase can only add nitrogenous bases to an already existing nucleic acid strand. So we start with RNA, and then the DNA polymerase binds and will actually then put down the DNA molecules. And the RNA primers will then be later cut out and actually replaced with DNA nucleotides. The ribozyme or spliceosome will be important in eukaryotic cells in that it actually will cut out and remove the introns that are the non-coding portions of the DNA strand and splice together the exons within the messenger RNA. RNA polymerase is going to copy RNA from a DNA template, so this becomes important in DNA transcription. And then the snRNP, topoisomerase, and transposase, these are also some other enzymes that we may mention as we go through some of this. So, DNA gyrase will undo the supercoiling ahead of the replication fork. DNA helicase will actually separate the 2 strands, and DNA polymerase then can come in and lay down DNA nucleotides matching T and A, C and G, etc. as we move down the parental strand of the DNA. Now, it's important because we're going to look at the structure of the sugar phosphate backbone here in a moment. And you'll notice that these 2 strands are actually made in opposite directions from each other. And that is because of the sugar phosphate backbone. So the initial processes in replication. DNA replication begins at an origin. And usually in bacterial chromosomes that are a singular circular chromosome, they have a single origin. But in eukaryotic chromosomes, because they are linear, we may have multiple origin points. This is an important point right here. DNA polymerase replicates the new strand of DNA, and I can also say RNA polymerase will do the same thing with RNA. only in the 5' to 3' direction. And that's why the two strands will be made opposite of each other. Again, let me say this. A new strand of DNA that's being made, or a strand of RNA that's being made from a DNA template, will only be made in the 5' to 3' direction. And because the strands of DNA are anti-parallel, in other words, they are flipped upside down from each other, each new strand of DNA during DNA replication will be synthesized slightly differently from each other and in different directions. The leading strand will be synthesized continuously towards the replication fork, while the lagging strand is synthesized discontinuously away from the replication fork. And what we will find is we have numerous RNA primers that will be necessary, and we actually will make the lagging strand in short segments known as Okazaki fragments. The Okazaki fragments are anywhere from about 1,000 to 5,000 nucleotides long. Then, once we take out all the RNA primers, we can use another DNA polymerase to actually put in the missing pieces of DNA, and DNA ligase will join the sugar phosphate backbone together. So, what we have is a 3' end and a 5' end. The 5' end will always start with a phosphate group. So let me get my pen up here and see if I can actually do this. So we're going to have phosphate here, PO4. The 3' end will be the deoxyribose sugar. So a 3' end will always start or end at a sugar, while the 5' end will always start or end with a phosphate group. When we make a new strand of DNA from each of these individual strands, the new strand will have to start at a 5' end. So on this strand, since it's 3', the opposite would be 5'. And so this strand will be synthesized continuously towards the replication fork. Whereas this one is a 5' end. So I can't start up here to 5' end and go this direction. So what I have to do is come over here to this end where it's a 3' end and start going in this direction away from the replication fork. But because I have to start with an RNA primer, I will butt up against an RNA primer out here, and so this would be the lagging strand that's made discontinuously in Okazaki fragments. So this just shows what I was talking about. So my 3' end here with my sugar. The opposite side of the new strand starts at 5'. We lay down an RNA primer. Again, later on, an enzyme will come in and take out the RNA nucleotides and replace this with DNA nucleotides, and ligase will seal up the sugar phosphate backbone. But we'll make this strand from the 5' going in the 3' direction towards the replication fork. But the lagging strand, I can't start out here with a 5' end because this already is. So I move over here where it's 3', and I'll lay down an RNA primer. The DNA polymerase then begins to lay down DNA nucleotides. But eventually what's going to and then I start back over here and make another Okazaki fragment. When this DNA polymerase butts up against this RNA primer, it gets kicked off. Again, then an enzyme will come along and take out the RNA primer, fill in DNA nucleotides, and DNA ligase will then actually seal the nicks in the sugar phosphate backbone. This takes a little more time, and that's why this is known as the lagging strand. So let's look at then the other thing that DNA is going to be involved in, and that is going to be protein synthesis. DNA directly synthesizes the direct synthesis, of enzymes and other proteins by ribosomes in the cytoplasm of the eukaryotic cell. Messenger RNA carries out the instructions encoded for in the DNA to the ribosomes of the cytoplasm, where it's then translated into a protein. So the transfer of genetic information is accomplished through transcription of DNA to messenger RNA, and translation, where polypeptides are synthesized then from the messenger RNA by the ribosome. And this is the central dogma of genetics, so that the DNA that holds the code for the protein will be transcribed to the messenger RNA, and then that will be translated by the ribosome to form polypeptides. The amino acid sequence then that is coded for by the gene in the DNA through messenger RNA will then determine how that protein folds up, and how the protein folds up determines its function. So here we have a strand of DNA, and I know it's DNA because I have thymine in it. One of these strands will be my template strand for making messenger RNA. If I'm going to start reading over here on this side, then which one of these will be my template strand? Well, again, when I make my messenger RNA, it has to start at a 5' end. So the messenger RNA will be read from the 3' end of DNA. And so T goes to A goes to uracil, because that's what's in RNA, C goes to G, G to C, etc., etc. And this is the process of transcription. In eukaryotic cells, this occurs in the nucleus. In prokaryotic cells, it simply occurs in the cytoplasm. Then the messenger RNA will be translated by the ribosomes into a series of amino acids. And a 3-nitrogenous base sequence of the messenger RNA, known as a codon, will code for each amino acid. So the codon AUG is going to code for methionine, CGG codes for the amino acid arginine, UAC codes for tyrosine, etc., etc. And the amino acid sequence then will determine how, again, the protein folds up. How it folds up determines its function, which determines the phenotype. So messenger RNA is synthesized from the DNA template. RNA polymerase is what's going to accomplish this. It will bind to the promoter site of the gene. This results in a complementary copy of messenger RNA. The RNA polymerase will detach from the gene at the termination segment, and we'll talk about how that happens. The messenger RNA then in eukaryotic cells has to first be processed, but will move out of the nucleus into the cytoplasm and bind to a ribosome, where transcription then will continue until termination sequence has been reached on the gene. So transcription is initiated at the promoter site of the gene. And so the RNA polymerase is going to separate or create a bubble within the gene or the chromosome at this point, at this locus. And this will then read the template strand of the DNA, making the messenger RNA copy. Once we have initiated transcription, we begin to match DNA nucleotide bases to RNA nucleotide bases. And this is then the process of elongation. And we're again making this new strand of messenger RNA from the 5' end to the 3' end. Again, simply matching T's to A's, A's to U, C's to G, etc. as we move right on down the strand. When we get to the termination or the terminator of the gene, this simply is going to tell the RNA polymerase we're done copying the material in the gene. And termination can occur by two methods in eukaryotic cells, either by self-termination, which we sometimes refer to as loop termination, or enzyme-dependent termination. Self-termination or loop termination is where we actually wind up in an area where we wind up with a lot of CGs, and literally the messenger RNA loops on itself, forming a loop, and that's what will kick the RNA polymerase off of the DNA. Or, in some cases, certain genes actually will carry along with them an enzyme that when we reach the terminator will separate the RNA polymerase from the DNA strand, and the DNA strand then will just zip back up. Now, once we have done RNA transcription in the nucleus of the eukaryotic cell, it will have to actually be modified. Transcription will also occur in the mitochondria and chloroplasts because they do have DNA. However, the genes contained within the small DNAs of mitochondria and chloroplasts are actually genes for the enzymes of energy production. And so again, these are not going to be replicated during cellular reproduction. So, in eukaryotic cells, three things will have to happen to the messenger RNA strand before it leaves the nucleus. First of all, on the 5' end of the mRNA, we will add a modified guanine nucleotide. This is known as the 5' guanine cap. We think that the 5' guanine cap may assist the ribosome in finding the first start codon found on the messenger RNA strand during translation. We also will add anywhere from 100 to around 250 adenine nucleotides to the 3' end, and this is known actually as the poly A tail. This may actually help stabilize the messenger RNA strand. The other thing is that in eukaryotic DNA, there are places within the genetic code that are non-coding for amino acids, and these are known as introns. They were once called junk DNA because we just said, well, they don't code for anything, so they must not mean anything. Well, we now understand that probably most of these introns are actually regulatory RNA structures that regulate gene expression within the cell. The exons will be the actual coding portion. So before we translate the messenger RNA to a protein, we have to remove the introns and splice together the exons. They are named because the introns, these little pieces of RNA, will remain in the nucleus of the cell. The exons that are spliced together, that are the coding portion, will exit the nucleus for translation. So the areas of the messenger RNA strand that do not code for an amino acid are introns and must be removed by spliceosomes or ribosomes prior to leaving the nucleus. What is left of the messenger RNA strand, the exons, will be spliced together to form the final or mature messenger eukaryotic RNA that will then exit the nucleus to go into the cytoplasm for translation. And these ribozymes or spliceosomes, either name, is a unique type of RNA that actually functions as the enzymes that will cut out those introns of the messenger RNA strand and splice together the exons. So here are the non-coding introns. And these enzymes will simply cut the DNA, taking out the introns and splicing together the exons, and we'll have that 5' guanine cap and the poly A tail, and then it can go out for translation. What most of the time these ribozymes or spliceosomes do is they actually form a loop of the introns, and then these loops will simply be cut out. We put again that 5' guanine cap, the poly A tail, and now we have the mature messenger RNA for translation. So translation is the process where ribosomes will use the genetic information that was found in the DNA in order to form the polypeptides of the proteins. Messenger RNA will be translated in a 3-nitrogenous base series known as a codon. Each codon will code for a single amino acid. Translation of the messenger RNA by the ribosome begins at the first start codon, which the start codon is the 3-nitrogenous base sequence AUG. Translation will end when the ribosome reaches a nonsense codon, and these are UAA, UAG, and UGA. When the ribosome reaches one of these, there is no amino acid that will be added at this point, and therefore protein synthesis will cease. Participants in translation include the messenger RNA, the transfer RNA, which is responsible for bringing in the amino acids to the ribosome, the ribosomes themselves, which are made up of ribosomal RNA, and of course then the 20 amino acids. And this table is a messenger RNA codon table that contains the codes for the 20 amino acids. Now, in some prokaryotic cells, they have a 21st and a 22nd amino acid, but we're just going to say for our benefit there are 20 amino acids. So, to read this chart, if you've never read one, which hopefully you have, but the first nucleotide base is read over here on the left side. The second nucleotide base in the codon is read on the top, and the third is read over here. So, AUG. So I start A and U and G, and you'll notice that AUG, not only is it the start codon, but it codes for the amino acid methionine. Methionine will be the first amino acids of all proteins. Now there's different forms in between eukaryotic and prokaryotic cells. I don't really care. Methionine is always going to be the first amino acid in the protein sequence. It may get lopped off later on by other structures within the cell, but the protein always starts with methionine. And you'll notice that UAG is the only codon for the amino acid methionine. Now, what if we had a codon of AGU? So A, and I go to G, and then to U, and that codes for the amino acid serine. What if I had a codon of UCA? So I go to U, and C, and A, and oh wait, serine. What? Serine? There are, if you count up all of these codons, 64 codons. 3 of them, right here, are stop codons, meaning they don't code for an amino acid. So there are 61, what we refer to as sense codons, that code for the 20 amino acids. So are we going to have redundancy within the code? And absolutely. In fact, only methionine and tryptophan have a single codon. Everybody else has 2, some have 4, and like serine actually has 6. That means that I could actually have an error in my code and not change my protein sequence. So if I had a change of UCU to UCG, I'm still going to get serine at that point in the protein. This is actually another form of the codon chart, and I kind of like this one just because I'm a visually oriented person. But you start in the center and work your way out. So the inner circle A, and then U, and then G, there's methionine start. Or again, if we had UCU, U, C, and U, that's serine. The multiple codons is responsible for what we call redundancy within the genetic code. Now, if I told you that my codon is U and C and I don't know what the third letter is, what is my amino acid going to be? Well, in this case, it's going to be serine no matter what the third letter is. And this is what's known as wobble within the code. Same thing if I had GUU. GUU codes for valine, but GUG also codes for valine. So this is the wobble. Now, transfer RNAs are also made up of RNA, and they are kind of folded up on themselves to make this kind of crazy clover leaf looking structure. There's two important things on the transfer RNA. One is right up here, and on the 3' end stem is the amino acid or acceptor stem region. This is where the amino acid will attach out in the cytoplasm to the transfer RNA to be transported or transferred to the amino acid or to the ribosome. Which amino acid attaches to the acceptor stem is determined by this 3 nitrogen base sequence right here, known as the anticodon. So if methionine is the one I want attached here, what I have to have is the complementary base pairs of RNA from the codon to the transfer anticodon. So since AUG is my codon, I would need actually A goes to U, U goes to A, G goes to C. That would tell this transfer RNA then to attach to methionine when it sees it out in the cytoplasm. The ribosome, made up of a large and small subunit, will come together over a messenger RNA, and it will actually then form 3 areas that we'll talk about here in a moment. The first AUG codon that is seen from the 5' end will be where we start protein synthesis. The first transfer RNA with the anticodon of UAC with methionine attached will also come in here, and so messenger RNA, the 2 subunits of the ribosome, and the first transfer RNA form what's known as the ribosomal complex. So here we start. Now watch very closely. 1, 2, 3, 4, 5, 6, 7, and now we have our protein. Cool, right? And this picture just puts it all together for you. So again, in eukaryotic cells, transcription occurs in the nucleus. We will then process the messenger RNA, taking out the introns, splicing together the exons, putting the 5' cap on and the poly A tail. It will then exit through the nucleopores of the nuclear membrane and will then attach to the subunits of the ribosome at the 5' capped end. It will then search out that first start codon, and this is where we then will start matching codons of messenger RNA to the anticodons and forming peptide bonds between the subsequent amino acids, forming our polypeptide chain. When we reach a stop codon or nonsense codon, this entire thing will then be released from the ribosomal complex and we will have our protein. As I mentioned before, chromosomes are not only found in the nucleus, but are also found in the mitochondria or chloroplast of those eukaryotic organisms. So how exactly are these inherited? Small amounts of DNA are present in these organelles. The mitochondrial and chloroplastic DNA contain genes that code for ATP-generating enzymes. Human ova contain several mitochondria, while sperm contain very few, if any, mitochondria, and the few that may be found tend to be non-functional. So all of the mitochondria that we have in our cells today came from mom. So they are inherited differently than genes on the chromosomes of the nucleus and are not transmitted from parent to child like the nuclear chromosomes. So this just simply shows a picture of a mitochondria. It is a double membrane bound organelle, and the inner folded cristae, if you remember, is where our electron transport chain will occur, and so this will actually be going on and producing a lot of energy for us. And these small circular structures are actually similar to the bacterial chromosome, and there are actually ribosomes that are in the mitochondrial matrix, and these actually are, interestingly enough, are 70S ribosomes, similar to what we see in bacteria. That's why we believe that mitochondria and chloroplasts probably were actually small organisms that the growing eukaryotic cells actually engulfed and caused them then to make the ATP for the larger cell. And so these actually then were small organisms that became the mitochondria. Hereditary diseases resulting from mitochondrial DNA mutations are inherited differently than genetic mutations carried on the nuclear chromosomes. Mutations of mitochondrial DNA typically tend to affect ATP production and nothing else. Transmission of abnormal mitochondrial DNA from parent to child is always from the mother. Very few rare cases have been seen of any parental mitochondrial DNA inheritance. So let's review a little bit about cellular division. Mitosis is the division of the somatic or body cells. Each somatic cell is diploid in number and contains 46 chromosomes in humans. Not all mature cells are able to undergo mitosis. So we have these what we call permanent tissues of cardiac muscle, skeletal muscle, and neurons that once they are mature cells and have reached full differentiation, they are arrested in the growth phase of interphase and actually stop dividing. Connective tissue and liver cells divide as much as they are needed, and in fact typically don't undergo a lot of mitosis unless they are replacing cells that were damaged from injury. Cells lining the testicular tubules that produce the sperm cells divide continually once puberty onsets. Blood-forming cells in the bone marrow divide continuously and are known then as labile tissues, along with epithelial tissues, to replace those cells that are lost in circulation or from the surface of the epithelial cells. In mitosis, there is no reduction in chromosomal number in the daughter cells. Each of the two daughter cells will actually be diploid cells. Each chromosome and its newly duplicated counterpart will lie side by side during mitosis, and these are known as the sister chromatids attached at the centromere. Each chromosome therefore duplicates itself through DNA synthesis before beginning cell division. Sequence of mitosis, if you remember, we have prophase, metaphase, anaphase, telophase, and cytokinesis, which occurs actually at the same time as telophase. During prophase, the chromosomes shorten and thicken as they are packaged tightly around the histone proteins. The centrioles are going to move to opposite poles and begin to form the mitotic spindles. Some of these mitotic spindles will attach then to the centromere of the sister chromatids, and the nuclear membrane breaks down. During metaphase, the chromosomes are all lined up along the equatorial plane of the cells attached to the spindle fibers. In anaphase, the sister chromatids separate at the centromere, and these then are pulled by the spindle fibers to opposite poles of the cell by the spindle fibers. During telophase, the nuclear membrane begins to reform around the now separated sister chromatids that become the chromosomes of the new daughter cells. The chromosomes begin to unwind into the chromatin form, and cytoplasm is going to divide by cytokinesis. What we'll see is that the cleavage furrow actually begins in late anaphase and continues through the end of telophase to divide the original cell into two genetically identical daughter cells. In meiosis, this is the development of the germ or sex cells. And this is where we get an intermixing of genetic material between homologous chromosomes. The chromosome number will be reduced to the N number, or the haploid number, during meiosis. It entails two separate divisions within the cells. The first meiotic division reduces the number of the chromosomes by half, so we go from 46 to 23 chromosomes. But before the second meiotic division, those then actually will replicate, so that in the second division we divide again, and each of those daughter cells will receive 23 chromosomes. So the daughter cells receive only half of the chromosomes from the parent cell in each of the division. The second meiotic division will be similar to mitosis, but each daughter cell will only contain 20. So gametogenesis, or the formation of the gametes, this is the process of forming these mature sex cells. And in the gonads, the testes and ovaries, they contain precursor cells called germ cells that will be capable of developing into the mature sperm or ovum. Spermatogenesis is the development of the sperm, and spermatogonia are the precursor cells in the testicular tubules. The spermatogonia are diploid cells, or 2N cells. The oogenesis is the development of the ovum, and the oogonial cells within the ovary are the precursor cells to the development of the ovum. Both processes have some similarities and differences. In spermatogenesis, the spermatogonia form primary spermatocytes by mitosis, and so these are going to be the 46 or 2N cells. They will then undergo meiosis, where we get primary spermatocytes form secondary spermatocytes, and each will have 23 chromosomes, or 23 pair. The second spermatocytes will form spermatids through the second meiotic division, and each one of these spermatids will now be a haploid cell containing 23 chromosomes. The spermatids then will develop into the 4 sperm. So from each spermatogonial cells, we will get 4 sperm. This is because we have equal cytokinesis in both meiosis I and meiosis II. In oogenesis, the oogonia form primary oocytes by mitosis within the fetal ovaries. These will be 2N cells. And during fetal development, these primary oocytes will form primary follicles and begin actually prophase 1 of meiosis, where they are then arrested until later development. The primary follicle, under the influence of FSH and LH at the onset of puberty, will mature one of these primary follicles into a mature follicle, and the egg will be ovulated each month. Now, the primary oocyte forms the secondary oocyte by the first meiotic division. And actually, meiosis I does not happen, or does not continue from prophase I, unless the cell is actually fertilized by a sperm. So, in other words, all of these primary oocytes have been arrested in prophase I and will not complete meiosis until they are actually fertilized. When and if this occurs, the primary oocyte will divide unequally and actually form a small and large cell. The smaller cell is known as the first polar body, and this is due to the unequal cytokinesis that occurs in the first meiotic division. The secondary oocyte, the larger cell of the production of the first meiotic division, then will begin a second meiotic division, and again this will be unequal cytokinesis, and we form a second polar body and the viable ovum. So in oogenesis, from one oogonial cell, we will only get one viable egg. So spermatogenesis, from each, again, spermatogonial cell, we get 4 spermatozoa because we have equal cytokinesis. Spermatogenesis occurs continually from the onset of puberty until actually death, and is carried to completion in about 2 months. Seminal fluid will always contain fresh sperm. Oogenesis, one ovum will be formed from each precursor cell, and the polar bodies will simply be discarded due to the small amount of cytoplasm that they receive during the meiotic divisions. Oocytes are not produced continually. Oocytes in the ovary form actually before birth and then again are arrested in that prophase 1 and will remain in that prolonged prophase of the first meiotic division of fetal life until ovulated and fertilized. So, congenital abnormalities from abnormal separation of chromosomes is much more frequent in older women, simply because the eggs are older and the chromosomes have been sitting in this arrested state for such a period of time that literally they become sticky. The ova released later in a woman's reproductive life have been held in prophase for so long, usually anywhere from a few years to 45 years, that the ova have been exposed to years of potential harmful radiation, chemicals, and other injurious agents that may have damaged the chromosomes. This predisposes to abnormal chromosome separation if and when the meiotic divisions occur, and this results in excess or deficient numbers of chromosomes or the loss of or addition of too much chromosomes. Now, single nucleotide polymorphisms, known as SNPs, these are actually important because these are due to errors within a single base that may alter the phenotype in the individual. We also now have the ability to recombine DNA from two organisms and come up with a unique organism pattern. We also have the ability now to do gene therapy, and this is a big boon. We may actually be able to replace deficient or abnormal genes with good genes. So, SNPs are actually structural variations in single gene nucleotides of different individuals. It can affect gene function, resulting in an individual difference in body functions or appearance, how rapidly the cell inactivates drugs or environmental toxins or repairs DNA damage. So it may allow for one person to be very susceptible to a disease while another one is not susceptible at all. Variation in responses to food, antibiotics, or drugs is oftentimes caused by SNPs, and the ability to detoxify potential carcinogens or susceptibility to cancer. Gene profiles determine the number of genetic SNPs or genetic susceptibilities to chronic disease and cancer. Recombinant DNA technology, this is oftentimes where we are using bacteria and placing in a foreign gene into the bacteria that produces a desired biological product, such as, for instance, human insulin is now produced actually by a genetically recombined E. coli, in which we have put in the human insulin gene into the bacteria, and the bacteria reads the gene and makes the insulin. So the recombinant DNA technology does have area of practical applications. It does help us increase understanding of the molecular basis of genetic disease by studying the normal gene structure and function and then altering it. We could actually then also diagnose genetic disease in the prenatal condition. And this is so that we could identify abnormal genes and gene products in the fetal cells and possibly actually even replace those abnormal genes then with good genes. This process requires insertion of a gene that directs the synthesis of a biological product, such again as insulin, into a bacterium through some sort of vector. And the vectors are either plasmids or bacteriophages, or we can actually put them in yeasts through vectors as well. Methods, recombinant DNA technology, we take genes from two different sources and recombine them into a single organism known as the recombinant. We have a unique genetic pattern within the recombinant. Genetic engineering is actually the manipulations of the genes already present in a cell or individual. And gene splicing is where we take a piece of genetic material, cut it open, and actually put in another piece of genetic material into that piece of DNA. Some products of recombinant DNA technology include a variety of things, and this is not an all-inclusive list, but certainly interferons, those chemicals that interfere with viral replication, interleukins, and tumor necrosis factor, all of which are involved in immune systems. Hormones such as erythropoietin, human growth factor, insulin, and others. Certain enzymes such as tissue plasminogen activating factor, or TPA, which is known as a clot busting drug, because this actually can dissolve potentially dangerous blood clots in myocardial infarction and thrombotic strokes. Certainly making safer vaccines. We could actually place in a gene for the antigen of a pathogenic organism into a non-pathogenic organism, allow that organism then to read the pathogenic antigen's gene and produce it in you, not causing disease, but actually causing an immune response to the antigen of the pathogen. So our vaccines have become much safer due to recombinant DNA technology, and vaccines for Hep B, human papillomavirus, and Haemophilus influenza type B, which causes a severe form of meningitis, have all been produced by recombinant DNA vaccines. And then things like factor VIII for hemophiliacs, or bovine growth hormone for giving to cattle to increase milk and or meat production. Now, gene therapy is an extension of the principles of recombinant DNA technology, but what we're really trying to do with gene therapy is fix the gene that is abnormal within a person or cell. So the normal gene inserted into a defective cell, let's say that lacks the gene for an enzyme or structural protein that will compensate for the missing or dysfunctional gene. The cells are removed from the patient, treated, and then reinfused, and hopefully will then basically grow within the patient and become normal or produce the normal product. Oftentimes, we're going to use viruses that carry the genes that are introduced into the patient's cells to actually treat these defective cells. So what we'll see is cells are removed from the patient. They are then actually infected either with an adenovirus or retrovirus that has been genetically modified to carry the gene that we want to implant into the patient. And oftentimes, actually, we take out the entire viral genome and put in just the gene that we want. The virus still thinks that it's got viral DNA or viral code, and so it will infect these cells from the patient. Now that viral code will actually be spliced into this cell's genome, and the cells then are re-injected into the patient, and hopefully they will then go to the areas where they are needed and begin to grow and multiply there. Most oftentimes this is done with bone marrow cells. Experimentation and clinical trials of gene testing have been carried out on human volunteers with particular genetic conditions. Most target cancers or single gene defects or infections. We now actually have some gene therapies that target the specific cancer in a specific cell in a specific patient. So while this gene therapy will not work with anyone else's cancer, even though it may be a similar cancer, it is so tuned to that particular genetic cancer that it will only affect those cells with that genetic profile. Most genes, again, are carried out by virus vectors, and primarily we still use HIV and adenoviruses as our primary virus vectors in gene therapy. Early trials were actually hampered by safety issues in which the viruses that were used as delivery vehicles actually caused malignancies. And part of the reason for that is early on, we just inserted the gene of interest into the viral genome and allowed it to infect the cell. So part of the viral genome was still present. Now we actually simply remove the viral genome, put in the gene of interest, and then put the virus in with the cells that we want the virus to, quote, infect. It still may actually have some problems because, again, we're using a virus as a vector. Germline gene therapy, where we really want to affect the genes, is actually within either the sex cells or early in the zygote. So we find the defect in the egg, the sperm, or the early embryo, and we actually inject or implant the gene that we want to function into those cells. This means the new gene is now present in all cells of that individual. And so the early embryo, the cells continue to divide, and each one of those now has a copy of that gene that was placed in the earlier cell. Therefore, the therapeutic gene is now inheritable. So germline therapy is actually a lot better than trying to treat just specific cells of, let's say, an adult. But how are we going to know? Well, we can actually look at the genetics of the sex cells or even the early embryo. But we're still in the beginning part of gene therapy. And are there some ethical issues that may go along with this? And there are, but we'll leave those to the medical ethicists. So this is the end of this chapter, and we'll pick up in chapter 4.